All right, so I'm going to dig into the lab. This is going to be a great review of everything that we've covered with regard to Mendelian genetics. Going to go through monohybrid, dihybrid, all the different types of complex inheritance, and some practice with pedigrees. It'll be a great review for your test, practice, and in your lab book there is an answer key, but that'll only help you verify your correct answers. If your answer is incorrect, it won't do much for you. So I'm going to go through and show you how each of these has worked so that you can learn where you're going wrong if that is the case. You can get help to work some of them if you're stumped. All right, so starting with monohybrid, we are told that cystic fibrosis is a recessive genetic disorder. So that means that they've designated in here, and the reason that they've designated in is because it could get confusing with C's. That's not a rule. You can still use those. You'll see people do the same thing with S's, but that's the reason why in a lot of cases a letter that has a different looking upper and lower case is used. So big N in this case is the unaffected or normal, and little n is for cystic fibrosis. The parental cross is homozygous dominant dad with a heterozygous mom. Set up our square. Put Ron there, Nancy here. And then it's asking for the ratios or percentages ratios for genotype and phenotype. You've got 50% homozygous dominant, 50% heterozygous, but since this is a recessive trait, 100% are normal or unaffected, which, to answer the question, the probability that one will have zero fibrosis, it's 0%. All right, the next monohybrid cross, freckles, homozygous dominant. So that means the big F is for freckles, little f, no freckles. The parental cross is Patty, who's homozygous dominant for freckles, Charlie, who's homozygous recessive for no freckles, and then the probability of the children. The probability of their children having freckles is 100% freckles, because 100% of them are going to be heterozygous, and the freckles is dominant. Right, the next one, Eddie has brown eyes, while Sybil has blue. Since then, they've learned that there's more to eye color, probably since this question was written, than just this. It is more recessive to have blue eyes than brown, but as you know, there's variation in blue, there's variation in brown, so it's polygenic to an extent. But blue eyes versus brown eyes, just generally speaking, is dominant recessive. So big B is going to be brown, little b is going to be blue. So I'm going to predict their offspring. Assume Eddie doesn't carry a recessive allele. So that means he is homozygous dominant for brown. If she has blue eyes, she has to be homozygous recessive. A 100% of their children are heterozygous, and they are all going to have brown eyes. All right, so that's the first page. We've got one more example of monohybrid, and in this case, looking at Huntington's disease, which Huntington's disease is caused by a latent dominant gene. Latent means it doesn't show up till adulthood. So this can be kind of tough because people don't know they have it until after they have had children in a lot of cases. So with dominant genes, since there is technically no carrier, usually it's not as likely to be passed on. But in this case, because even though there's dominant, it's dominant, it doesn't express or show up as a trait until after reproductive age, or at least till you're past the age where you become reproductive. So in this case, big H is Huntington's. Little h would be normal or unaffected. And let's see, the father is heterozygous, wife homozygous recessive, and Punnett square for the probability the children would inherit the allele for Huntington's. Let's put him up there. Sorry, that's kind of hard to see. I think I can make my point of this pencil smaller. Let me do that. Oh, maybe not. Oh, this is a pointer, not change the size of the point. Sorry, this ought to be entertaining. Oh, here we go. I figured it out. Yeah, that's better. All right, so 50%, because here and here we've got the allele for Huntington's. So the probability that the child would inherit the allele for Huntington's is 50%. Okay. All right, so now we'll move on to the DiHybrid cross. And remember, this is looking at two different genes. So in garden peas, we're going to be in the peas again. Tallness is dominant to shortness. So big T is tall, little t is short. And axillary flowers are dominant to terminal flowers. Oh, no, I can't do that. All right, so now looking at the expected ratios for the genotypes and the phenotypes. If a heterozygous tall, heterozygous axillary plant is crossed with a heterozygous tall terminal plant. Okay, so the heterozygous tall, heterozygous auxiliary would be crossed with a heterozygous tall terminal plant. Now, remember, for this, we're going to come up with our different gamete possibilities. So I've got 4 possibilities in each instance. Say this is the male, although in this case it would be sperm, pollen, but anyway. So I've got this possibility, this possibility, okay. So really only 2 different possibilities. In this instance, there are 4 different possibilities. All right, so now I can put those over here. Then I'm going to do my cross. I don't know why I wanted to write those small. All right, so now looking for the genotypes and the phenotypes. Okay, well, let me look for all right, so genotypes: homozygous tall, heterozygous auxiliary, got 2 out of 16. Oh, sorry for the weird lull there. My Apple Pencil stopped working and I had to pause and charge it. Okay, so where were we? I had just done homozygous for tall and heterozygous for auxiliary, and the phenotype is tall auxiliary. All right, so now let's see. I'll look at the ones that are, oh, I had started that heterozygous for both. And that is 4 out of 16. And the phenotype is the same. Now, how about homozygous at both? 1, 2. And in this case, it is tall, and the flowers are terminal. Heterozygous tall with terminal flowers. 2. 4. Oops. And that is the same. And then I've got 2 for homozygous short and heterozygous auxiliary. Sorry, I'm now writing over here. And 2 for homozygous recessive at both. And those are going to be short, and the flowers are going to be terminal. So now let's look at our ratios for our phenotypes. So we've got 6 out of 16 that are tall and auxiliary. 6 out of 16 that are tall and terminal. And 2 out of 16 that are short and auxiliary, and 2 out of 16 that are short and terminal. So those would be our genotype and phenotype ratios on that DiHybrid cross. All right, so here's some more practice DiHybrid problems. In horse color, black is dominant over chestnut. So make the key. And trotting gait is dominant over pacing gait. Homozygous black pacer. So since it's homozygous, I know I'm looking at this. Made it to a homozygous chestnut heterozygous trotter. What will be the ratios for genotype and phenotype in the first offspring generation? So we've got to look at the gametes here. All the gametes for this one are going to be big B, little t. That's the only possible combination. And these are going to be 50% this and 50% this, because you're going to get the same either time from here. So now if we make our big square, all right, so, oh, well, I wrote the B in, let me switch that. I don't know why I did that way. I wrote the B in front in the one instance, and the T in the front in the other. That's going to make it, it's not technically incorrect, but it would definitely be easier to navigate if it were consistent. So I'll rewrite those. All right, so big B. All right, now you've really only got two different things going on here. So all of these are the same, and all of these are the same. So we've got 50% heterozygous, black, trotter, and the other 50% are heterozygous black pacers, because it's homozygous recessive. All right, just a couple more of these DiHybrid crosses. They can take quite a bit of time. All right, so we've got a black dominant over brown for rabbit coat, and short hair dominant over long, and the cross between a heterozygous black, heterozygous short-haired male. Oops. Put my X in early. Crossed with a brown, long-haired female. So she would have to be, in order to be brown and long-haired, she would have to be homozygous recessive at both traits. So she can only generate one possible gamete combination, and he can generate several. All right, so let's make the square. BS, BS, BS. A bunch of BS. That's kind of funny. All right, let's go ahead and work this square. All right, so our ratios for genotype are in rows, so 25%. So right here, this row, 25%. So 25% or 4 out of 16 for each of those different possible genotypes. Now for phenotypes, this 25% is going to be black and long-haired. This 25% is going to be black and short-haired. This 25% is going to be brown and short-haired. And this 25% is going to be brown and long-haired. So in this, in this case, the ratios are going to be the same for phenotype and genotype. All right, and I think this is the last of the dihybrid. It says, imagine a couple is planning to have children. The male is heterozygous for Huntington's disease and homozygous dominant for Tay-Sachs. So we already know that Huntington's is dominant. The couple is curious about the possibility and probability of their offspring inheriting Tay-Sachs and or Huntington's. For humans, Huntington's disease is dominant, and the normal condition is dominant in Tay-Sachs. So, oh, you know what? Let me go back. It asked here why N was being used. It isn't because of the C's getting confused. It's because the N is normal. Huh. I actually didn't think about that until now. So that way you know that the normal is dominant. I will not be putting that on a test since clearly I can't remember myself. Okay, and then the small n would be Tay-Sachs. Okay, so let's see. What did it say the parents were? The male is heterozygous for Huntington's and homozygous dominant for Tay-Sachs. So that would look like this. Oh, I did it again. I keep wanting to put the X too soon. And the female is homozygous recessive for Huntington's and heterozygous for Tay-Sachs. All right, and they want to know probability of genotypes and phenotypes. So our different allele combinations. We've got two possibilities here. And two possibilities here. So now we can do our square. Oh, those are really not straight lines. All right, and so let's do this. I had to make my pencil tip even smaller. This is the last of the dihybrid crosses. Thank goodness, they take a while. Oh, I botched that last one. Oh, shoot. All right, so then for our genotype and phenotype ratios, let's look for all the heterozygous at both first. Okay, so four out of 16, and that is going to be Huntington's and normal for Tay-Sachs. Okay, so then heterozygous Huntington's, homozygous dominant normal. It's also 4 out of 16. And that phenotype is also going to be Huntington's normal. So homozygous recessive for the Huntington's gene, which means normal, and homozygous for the Tay-Sachs, homozygous dominant, which means normal, 4 out of 16. Normal for Huntington's, and heterozygous normal for Tay-Sachs, 4 out of 16. So that's the genotypes. For the phenotypes, you're going to have 50% or 8 out of 16 Huntington's normal. So they're going to have Huntington's disease, but not Tay-Sachs. And then the other 50% or 8 out of 16 will be non-Huntington's and non-Tay-Sachs. All right, that's the last DiHybrid cross, thank goodness. And I am actually going to stop this video here because I'm afraid if I make it too much longer, I'm not going to be able to upload it on YouTube without paying for an account, because I think that I've just about hit the limit. And so I will upload an additional video that will start with sex linked.